Good afternoon, everyone. Welcome back. And we were discussing about cDNA libraries. We have discussed about genomic libraries before, and today we'll continue with cDNA libraries. In the last class, we have discussed two of the protocols, the last ones at least, or let's get back a little bit, and then we'll be able to see some of the points there. So when we were discussing about cDNA libraries, we had come across some of the problems. The problems are, one of it is with reverse transcriptase. Maybe it cannot reverse transcribe completely because its processivity is less. Sometimes there are truncated mRNAs. That is one problem. Using S1 nuclease is a problem because when it digests the hairpin loop, we will lose some of the information. And we also want the cloning to be in a proper direction if we want to express the genes that we are cloning. And in some of the mRNAs, maybe the hairpin loop does not form, because of which second cDNA strand synthesis might become a difficulty. So there are a lot of issues with that. Some incomplete or truncated mRNAs is also a problem. So when we are trying to learn about many different protocols of cDNA libraries, some of them will address one or the other. So we came across in this protocol at least, where it is solving at least two problems. The first problem is we are not going to use S1 nuclease, because of which it's a good thing, because we are not going to lose any information from the 5' end of the mRNA. We have also learned about two other protocols using oligo-DT. One of it is the same as what we have discussed a little while. We are not using S1 nuclease. And then if you're not using S1 nuclease, or first of all, how are we synthesizing the second cDNA strand? Second cDNA strand is synthesized by using homopolymer tailing. If you want to synthesize cDNA double-stranded, we should have known the information because we are going to use polymerase. For the first cDNA strand, we are using oligo-DT. And because we know the information here, we know that it is a poly A tail. But we don't know what is on this side because it is different for different mRNAs. So by using terminal transferase and DCTP, we are able to tag on both the sides. But when we use alkaline conditions, the mRNA is broken down. And now we have the first cDNA strand synthesized. We know what is on the 5' side because it's the complementary one. And we know the 3' side as well of the cDNA. And therefore we can use a oligo-G DG as a primer to synthesize the second cDNA strand. So we are not using a S1 nuclease, so we're likely to get more information, or we are not deliberately losing any information. Here is a protocol in which it is using S1 nuclease. That is a defect. But this is at solving the problem of directionality. We want to clone it in proper orientation so that the transcription would occur and then protein would be produced. So for that to occur, it should have been cloned in proper orientation. So directionality is the objective here. So what we should know is, if we want to clone it, assume if there is a vector which is having a promoter like this, and it has the EcoR1 site and SAL1. We want to put the fragment here. If we want to do that, then the gene that we want to clone here should on one side have EcoR1, and the other side it should have SAL1 overhangs. So how can we do it differently? So after synthesis of the second cDNA strand, first cDNA strand is synthesized, we add DNA polymerase and synthesize the second cDNA strand. Or it is still a single strand DNA because its phosphate backbone is complete. Only thing it is base paired. And during this stage, we can add SAL1 linkers on one side. It will only get added to one side because there is nothing to to ligate here, it is still complete. And then we are treating with S1 nuclease. So we now have double-stranded DNA to which we can add the EcoR1 over linkers, which could get added on both the sides, polymers of them. And then we are going to cut it with Sal1 and EcoR1. We will get a fragment, which on one side has EcoR1 site, on the other side it has Sal1 site. And that could be cloned into a vector easily. Right, this is the summary of what we have learned. We are going to learn a few more topics or few more protocols trying to solve. So see the logical flow of the steps and try to understand or try to remember what is the objective, right? Like I'm trying to describe here, the objective is not to use S1 nuclease and still be able to get double-stranded DNA. How is it achieved? That is the objective. And how is it achieved? By using homopolymer tailing. And in the other one, the other protocol, the objective is directionality. How is it achieved? Is by treating with or adding linkers at different steps, so that on one side we get EcoR1, on the other end we get Sal1 overhangs. And so there are protocols. What we are going to do here is we want to have, this is a nice protocol as well. It solves the problem of directionality. And the second thing is to get complete second strand or complete DNA, or not use, we don't want to use S1 nuclease. Okay, I hope you understood that we don't want to use S1 nuclease and still be able to synthesize the second strand. So here is the protocol. In this, they have made an improvement here. We are using, this is the mRNA with the poly A tail, and then we are going to add oligo DT. That means it has multiple T's with a Sal1 in the incorporated into the primer. So it's a Sal1 linker. Like you remember when we discussed about designing cloning primers, what do? We use a region of homology. This is the 5 prime, so I'm putting like this, region of homology. And then we have the restriction site. And you can call it as the linker there. And we have some buffer sequence, which is used for increasing the efficiency with which restriction enzyme binds and recognizes it or cuts it. For cloning primers, we have done something like that. In a very similar fashion, here they have chosen a oligo DT along with a Sal1 linker. So on one side, when the first cDNA strand is made, you're going to have a Sal1 link, Sal1 site here, and the first cDNA is synthesized. We can remove the RNA by alkaline lysis and so on, alkaline changing into alkaline conditions, and then we're going to add DNA polymerase and random primers. Random primers are something like this. They could be very small ones, something like hexamers, and it's a mixture of hexamers. Okay, so for different organisms, people calculate make a mixture of these hexamers. Hexamers can be of several combinations. So GATCAG. This is one combination. It could be, another one could be something like this. So what you are actually getting is what you are having as a random primers is a mixture of these hexamers. You're going to use them as primers. These are oligonucleotides, DNA, and they are 6 bases usually, and they are a mixture. They're not, it's not one constant sequence, right? I hope you understood. You can have any of those made sequences like this. So we add the random primers and then add Klenow fragment, which does not have the 5' to 3' exonuclease activity and the DNTPs. So in different classes, or say this is one mRNA and this is another mRNA, each of these mRNAs may have a sequence. One of the primer can bind, different primers can bind the mRNA in different locations like this. Different RNAs can be located because the probability is, say, about 4 kb. Every 4 kb, there is a probability of formation of a particular occurrence of a particular sequence. So what happens is that when we have such situation and we add clino fragment, it will synthesize and will drop there, will stop there, will synthesize and will stop there. So that way, we are likely to synthesize the whole cDNA, the second strand. This is what we are synthesizing. And then, of course, it will have oligo A, and also when polymerization occurs, it will happen like this. The key or the central to this is about the diversity we have in the random primer sequences. The more of these random sequences we have, the majority of it is possible. And I would want to, before going ahead, I would want to speak a little bit about how DNA synthesis actually works. And it will come also, it's not directly part of our work, but our class, but I think it's nice to learn about it as well. Say this is a stationary phase. I'm not discussing exact every detail, technical detail, but I'm trying to describe the principle of how it is done. So I have a stationary phase to which I am binding a base, say G, and it has OH, 3' OH. So now I'm going to say, for example, I want to make a sequence like this. Okay, I want to make a sequence like this. How do I do that? Chemical synthesis. So the next step I want to add is A. So I should have taken DATP. I'll just write ATP for ease of writing here. So we'll take ATP. And what will happen to that ATP? It has here is the 3' OH, to which the 5' side of it should bind here, 5' phosphate, and the pyrophosphate should be released out. ATP will have, I will just put it like that. This is the, one second. Sorry, it doesn't work. That is adenine there. And then what we have here is deoxyribose. And this is the 3' OH, right? And here, okay, I'll put, sorry, this is adenine. And I'll put it here. This is the 5' side, CH2OH, and and then we have the 3 phosphates. Okay, that is the simple way of representing ATP. So what should happen now is there should be a bond between these two, the alpha phosphate and the 3' OH of the base that is already here, right? And then that is when we would have gotten these two sequences, A, G, and after that A. So I will pass in a solution of containing ATP and activate reaction, so such that the 3' OH is going to make a phosphodiester bond along with the phosphate here. Okay. But then when we activate this OH to perform something like this, the 3' OH also will add. And there are many other ATPs around. And then those ATPs might bind here also. We will instead actually get GAA, multiples of them. That is not what we want. We don't want that. So somehow the 3' OH that is present here should be blocked from participating in polymerization reaction. So through chemical means, they would have actually blocked it. They would have blocked it so that it will not participate in any further reactions, any reactions at all, of the only this one, ATP, right? So now what we have, there is only one reaction that can happen, that is between G, the 3' OH, and this phosphate only. So all the G's that are present here, say for example, the same one is what is happening to it, we will look at it. G is now linked with A, like that. Now, after this treatment, we would have washed the column or removed all the ATP unbound ones, everything we would have removed. And now we have to synthesize something else. We need to add a base, and that is the T. So what we will do is we will add TTP, which is on the 3' OH is blocked. Okay, I'm just discussing the principle of how people do these things. Now, and then they will have activated the 3' OH. The one that was actually blocked here is now activated, and the linkage will, covalent bond will form like this. So it will become GAT. Now they would have washed it again, and then they would have supplied, because another T has to come, they would have given another TTP, and that would have added T here. So that is how they slowly build the nucleotide. Everywhere when we have primer synthesis and many other oligonucleotide, the principle at least will be of this kind. Okay. Now, why I'm discussing about it is we are supposed to use about random primers. How can we make it random primers? We want 6 bases. Random primers means we want a diverse condition, diverse bases. It could be any of them, any several different combinations. How much would it be? It would be 4 into 4, 4 to the power 6. What would that be? Anybody? Yes, anybody? Can you calculate? You might be having a calculator around.
4096.
4 to the power 6 is 4096. So probably we need to have somewhere around 4096 combinations of hexamers. How do we get that? So we'll use the same principle. The principle will remain same. And what they would do here is they would have run 6 reactions that we discussed just now, 6 of them. What they would do is, say they would have taken any of the bases with the 3' OH, say for example, a combination of them. Then they would add D and TP. All of them, their OH is blocked, not allowed to participate. So we would have added, in one case, we might have, because we are taking a mixture of bases. So in one case, G would be added, in another case, C would be added, in another case, T, another case, A. And then washing step. Then again, they would have put DNTP and allowed the reaction to happen. So you will have several types of reactions because you're you're using a mixture of DNTPs. So anything can be added randomly with anywhere. So we are actually generating a diverse mixture of hexamers. And then there would be, if we have an mRNA, it can bind, the probability that it will bind somewhere is very high. And if there is a gap between a primer and primer, we are using Klenow fragment to fill up these gaps. So it will fill up till here and it will stop. So we might have discontinuous ones, but we will have something like this, right? The second, this is for discussing, we are discussing about the synthesis of second cDNA strand. Okay, that is how random primers can be useful. Yes, and in some cases, random primers can also be used in multiple other places where, say, for example, here we are having convenience with working with eukaryotic mRNAs because we have poly-A tail. What about bacterial RNAs, bacterial mRNAs? They do not have poly-A tail. how do we synthesize the first cDNA strand to start with? There too, random primers can work very well. Random primers along with reverse transcriptase and so on. We will discuss that in some other time. I hope you understood how random primers can be used. And once we have synthesized, we will get SAL1 here. And later we will use EcoR1 linkers. So we'll have EcoR1 linkers added on both sides. But SAL1 would have cut here and EcoR1 here. So we'll get a double-stranded cDNA with EcoR1 and SAL1, like that. Okay. So there are limitations with um, several of those, because this is when we use oligo DT, we have 3' end biased. That is what we just was discussing. If something didn't have poly A tail, then they would not be cloned at all, right? And there, these random primers can be used. So we are still not done. We have problems to speak of, address, is that if we use these, our reverse transcriptase may not synthesize very long mRNAs if they are very long. Or in mRNAs, some of them could be truncated. The 5' G cap is broken off. But this will be synthesized too. We don't want incomplete ones like these. The following protocols, at least two of them, are to address both these situations. How to get full-length mRNAs. We don't want any of these half mRNAs or truncated ones. This is called as capture method. It is based on the fact that eukaryotic mRNAs have the G cap. Right? On the 5' side of them, they have the G cap. On 3' side, nothing. So the first cDNA, here is the protocol. We will just go through it and assume that there are, this is to solve where reverse transcriptase may not be able to synthesize the whole length, right? Especially if the mRNA is very long. How to get rid of incompletely reverse transcribed. We want to get rid of incomplete reverse transcription or reverse transcribed first cDNA strand. Okay, we want to avoid those things. So here is the mRNA. We will synthesize the first cDNA strand as usual. We can use the oligo DT and synthesize. In this scenario, and on this side, we are going to see, assuming that the reverse transcriptase is able to synthesize until the end, complete synthesis. Whereas on this side, there is incomplete synthesis of mRNA, or reverse transcription of incomplete reverse transcription of mRNA. So reverse transcriptase only synthesized till there, and this region has not been transcribed or reverse transcribed. So to the reaction, we would add RNase A. RNase A is single strand specific. Wherever it is, it will find single stranded RNA. It would cleave that. So in this case, the RNA is completely bound like this. So there is no degradation by RNase. The RNase cannot act on it. Whereas in incompletely reverse transcribed ones, this part of it is not base pairing, so it will be degraded by the RNase A. And now we're going to use affinity chromatography using EIF. E is for eukaryote, IF is for initiation factor 4E. If we remember or recollect in a transcription, translational initiation, the mRNA is recognized by one factor, that is EIF4 for E. So it has high affinity for the G cap. Now, when we pass this through the column containing the stationary phase containing the G EIF4, okay, So here we have this EIF4E, and we pass the both the mixtures, this one as well as this one. Which ones of these two have GCAP? Is there is no GCAP in the incompletely synthesized ones because because of the RNase treatment. Whereas the fully, wherever the reverse transcription is complete, those will retain the GCAP, and they will bind to the column like that. The mRNA will bind, and this is the cDNA that is synthesized. And once we have washed the column, we will elute the cDNA along with the mRNA, and then we will perform alkaline lysis to degrade the mRNA, and using random primer also we can synthesize the second cDNA strand like in the previous protocols that we have chosen. Here, I hope you understood the point, essence of it. Is the objective of it is to get rid of incompletely reverse transcribed cDNA, right? And we are using the concept or the point, the affinity of EIF4E for GCAP. And the incompletely transcribed and completely transcribed, we can get rid of this cap for the incompletely one by using RNase A treatment. Wherever reverse transcription is complete, RNase A cannot act upon it because of the double strand, because of the base pairing like this. Okay, this is called as capture method. You can also use it, assume it has two meanings at least. You can say for GCAP, 5' GCAP is one way, or the way you can remember is that EIF4E is capturing completely reverse transcribed those mRNAs with the 5' cap. Okay, you can associate the principle with the title so that you can remember what it means. Okay, so here is another protocol. This protocol is to eliminate truncated mRNAs. So if you remember that mRNA are produced, right? Gene transcription, there is splicing of exons and then mature mRNA. So mRNA is produced, it is translated, but mRNAs will also be degraded. This is how it is represents degradation. And some of the mRNAs could be broken, truncated also. And we don't want to have any cDNA of these truncated ones. How do we eliminate them? The more we have, why are these kinds of protocols very important is it will increase the library size, and most of the clones may not be useful. And as we will see in the following slides, when we screen, maybe through hybridization, they might give a hit that this one has the sequence. But after trying to characterize, we might fail it at the end because maybe we have cloned an incomplete cDNA. And that's why it's important to eliminate these incomplete cDNAs. So this technique, this protocol is called as oligocapping. So let's go through it. Here are two mRNAs. First one is a full length. How do we distinguish what is full length and what is not? Is simple. Is on one side we would have had poly A, on the other side we will have the GCAP. That is a full length. The partial length ones will miss either of them. Either they may miss GCAP or the poly A tail. Here we are only concerned, we are writing only poly A tail because for the first cDNA strand, we usually take the oligo DT as a primer. So that is the only thing that makes sense to us at this point. If we are doing reverse transcription, that is the only thing that will get transcribed. So somehow we don't want any cDNA forming from these partial or truncated mRNAs. How to get rid of them? So we are going to treat this all the mRNA that we have, the isolated, and then we are going to treat it with bovine alkaline phosphatase, And what it does is, I hope you can also recollect that the G cap is something like this, G, and then there is a phosphate. There are 3 phosphates, and then you have the mRNA. Okay, there are 3 phosphates. This alkaline phosphatase will act on these phosphates and will break it off like this. So what you will be left with is a kind of OH. So normally we would have add 5' phosphate, and for mRNA we have eukaryotic mRNAs, we have a G cap, and now the G cap is removed. So let's follow through what is going to happen to both these things. In the first one, we have sorry. In this case, we have G cap and poly A, that is a full-length mRNA. In the other one, the partial one, the cap is missing, poly A tail is there. Using alkaline phosphatase, we are able to, sorry, I missed it. So alkaline phosphatase is specific for, I made a mistake, okay? Sorry. So alkaline phosphatase is going to remove the phosphate alone. I mixed up with the acid pyrophosphatase one. Okay, let me redo it. So we have cap, and the truncated ones will have the 5' phosphate. And when we treat with alkaline phosphatase, the phosphate group will be removed, right? We remember what alkaline phosphatase does in during cloning as well. And then we are going to treat. So the phosphate is now become, the 5' phosphate has become 5' OH. The cap protects the 5' end of this full-length mRNA from the alkaline phosphatase treatment. Then we are going to use acid pyrophosphatase. That is right. Pyrophosphatase is, I hope you remember, pyrophosphate means you will have at least 2 phosphates like this. In GCAP, we have 3 phosphates. So the pyrophosphate will break the bond. Where is it? It'll break the bond here like this. And these things, the GCAP will be eliminated. Okay, what you will be left with is a 5' phosphate. So again, follow it. In these 2 cases, what is the 5' end? Here we have a 5' end in the top one. When it is still in this level, we have a G cap, and this one has a 5' phosphate. Upon treatment with alkaline phosphate, will cap truncated OH 5'. Then treating with acid pyrophosphatase, the full length ones, the cap will be removed and leaving a 5' phosphate. And the truncated one has 5' OH. And now we are going to add, as the title says, oligo capping. We are going to add an oligonucleotide to cap both of them. But then for ligation reaction to happen, okay, if we want ligation reaction to happen, we would need a 5' phosphate, right? If you remember, and we need a 3' OH. And only then you will have a phosphodiester bond forming between these bases. So if we see in the full length one, which upon treatment with the acid pyrophosphatase has cleaved and left at 5' overhang, 3' phosphate on the 5' side. The truncated one has OH. So let's try adding an oligocap. Oligocap is just a nucleotide and trying to use a ligase. It is a RNA ligase. Say, for example, I'm adding GAC, and this one has 3' OH. Can ligation reaction happen? Let's think about it. Similarly, we will see GAC and 3' OH. Okay. Can ligation happen between OH and OH? It cannot happen. Whereas the oligo, assume this is the oligo, and the OH and phosphate, yes, ligation can happen and form. So the full length mRNA would get oligo capped. This is the oligo that has been added. Whereas the partial or the truncated one will not get any oligo, because we know the 3' end. We will synthesize the first cDNA strand by using oligo T. Okay. And for the now we also know the oligonucleotide, this cap. We know the sequence of it. So we can use the complementary strand to the oligo and synthesize the second strand. Okay. Let me elaborate it a little bit. Let me try. So let's try to follow what is given here. I'm trying to talk about the primers. So here is a full length one, and we have A, or maybe I should have put it, just excuse me. So here is the full length one, okay, and already treated, and the oligo cap is there. I will, for ease, I will try to put a sequence here instead, so that it will be, you can follow it. As I said, I'm using GAC, so I'll just continue with that. GAC is the oligo that has been added. There is other mRNA, the truncated ones, which have these, but no oligo, because the oligo did not get added on. And here it is a RNA ligase. Please keep mind that also. This is a new enzyme that we haven't discussed or touched upon so far. So to both of them, we are going to add oligo DT. So, and then allow the reverse transcription to occur. So both of them will synthesize the strand. What will it be here? GTC, right? So this would be the 5' side, and here is the 3' side, 5' and 3'. We will then, using alkaline conditions also, we can get rid of the mRNA. The job of mRNA is done. We now have cDNA. Some of them are full length and some of them are partial, truncated ones. We want to eliminate these. Now, if I perform PCR, I would have designed primers. The primer would be GAC. This is the 5' side. And I would use another primer also, that would be oligo DT. Say, if I'm just adding primers, assume like this, GAC, and trying to do PCR or synthesize the second strand. The second strand will be synthesized in this case like this. But there is no GAC or the primer, the primer that we have added to the primer specific to the oligo is not present here, or the oligo is not present. So this will not, second cDNA strand will not form in this case. It will form only here. Okay. I hope you understood this. If you have any questions, please let me know. I will try to discuss it differently if you have any doubt in this protocol. Anything here? So I'll just quickly summarize. So the objective of this protocol is to get rid of all the truncated mRNAs. And in that case, we are first, the differences between truncated and full length is, full length one has both the GCAP as well as the poly A tail, whereas the partial one does not have the GCAP. By treating with alkaline phosphatase, we can remove the phosphate from the 5' ends of the truncated mRNAs, while the full length one will be unaffected because of the GCAP. Then we are going to treat it with acid pyrophosphatase. Acid pyrophosphatase acts on the GCAP and removes the G with two phosphates like this. It's breaking there. So they will be removed. And what will be left is the 5' phosphate group. The difference between these two is one of it, the full length one, has the phosphate group on the 5' end, and the truncated one has OH. Then we are going to add an oligo, any sequence that we know of, and then using RNA ligase to add to it. And then the full length ones will get capped or attached with the oligo because it has a 5' phosphate, which is essential for making the phosphodiester bond. Whereas in the truncated one, it has OH, and therefore it cannot form a bond with the 3' OH. So the full length ones will get capped while the truncated ones do not get capped. Because we know the sequence that we are adding, we can always have synthesize design primers accordingly. I mean, here in this case, the first cDNA strand is synthesized using oligo DT. Both of them will be, first cDNA strand for both will be synthesized. But the second cDNA strand will be synthesized only for the full length one because we know the oligo, the sequence of the oligo that we have added. So we can always try to take the design primers based on that and synthesize the second cDNA strand for the full length ones. And then we'll go through the cloning process. Okay. Any questions here? You can ask me so that I'll try to discuss differently if required. Okay, we'll move on. You can always message me or ask me in the next class if you want. So let's compare what is with cDNA library and genomic library, advantages and disadvantages. So in cDNA, the genomic library, we already discussed this. It contains the sequences of the entire genome. That's a good thing. If you want to know about the whole fullness of the genome, then that is how we should do it. We should have known each of the genes, the intergenic sequences, the regulatory sequences. They are only possible when we make a genomic library. When we make cDNA library, the focus is, or whatever is there, is only those that of the mRNA molecules. cDNAs are made from there. So it is only focused on the coding regions. And that too of the regions that are protein coding regions. Right, majority of them. Because non-coding RNAs, tRNAs, rRNAs, the information of those will not come here. The library size is usually small because it's only about protein coding sequences, whereas this size, when you say library size, that means number of clones that we need to make. Okay. In second thing is, in this case, the genomic library, the genes also have introns. Those intron sequences can also come up in the genomic library. Whereas in the case of cDNA library, they will be lost. It's an advantage in one way that we can clone a complete protein. We have sequence required to generate a complete protein without any interruptions. But we don't have the information for the introns. In this case, we have information for the introns for the genomic library, but we cannot express them as it is in every organism, because that organism should have compatible splicing activities for recognition and so on of the intron exon junctions, and then splicing to occur properly. That is another important thing. Yes, though I already discussed lack of introns. So mapping chromosome is difficult. And yes, these are that we already discussed the libraries. It is, of course, each of them have their, based on the objectives of the lab, we choose whether we want to go for cDNA library or genomic library. Both are important. It all depends on the objective of our work. Okay. I want to discuss something about screening. I think in one of the classes I discussed about what is meant by screening and what is meant by selection. In screening, in selection, if you have some 100 clones and you want to select only the one that is, say, for example, antibiotic resistant one, we would have plated all of them on plates containing antibiotic. Only those that are resistant would grow and the rest would die, right? That is selection. We are selecting means we will allow the growth of only those that we require and not the others. Screening is like something else. Screening is you take 100 clones, and when you plate them, they should have some characteristic different, because of which you can identify, yes, this is the clone that I want. So you already have clones or things, and by visually or some experiments, additional experiments, you can figure out which is the clone of interest, right? You can simplistically think of screening as something like that. So now we have generated, say, for example, cDNA library, a huge library, or even genomic library. How do I get the gene of interest from it that I want? Right? We want to figure it out. We also discussed about amylase gene and so on, how to the series of steps that we should follow when we want to isolate that. So I will come to this some other time. Remind me, PCR as alternative method to cloning. It's an interesting paper that was published in 2013 or so. Now we are trying to screen, how to screen. That is what we are going to learn. We have several techniques in it. One of it is by colony hybridization. The other ones are by immunochemical method, and then there are also insertional inactivation and so on. The simplest one is, say, for example, you have made a this is the marker, okay? This is the vector alone, and this is the test you can think of. Say, for example, I want to know, I have performed, there is also one more called as colony PCR. I will describe. It's nothing, it's simple PCR, just simple PCR with little bit of modification. So what we do is we perform ligation, assume that. When we perform ligation, assume there is religation also, vector-vector religation. And here, this is vector fragment ligation. We want the recombinant. This is the recombinant. We have transformed it into E. coli. Okay. Say, for example, these are two E. coli, and one of it has the vector plus fragment, and the other one is vector-vector. How do I know which one is recombinant and which one is not recombinant? Assume there are no other ways of trying to know. I should be somehow able to figure out the recombinant clone. So in that case, what we can do is isolate the plasmids from both of them. And then you can do simple PCR, or you can do restriction digestion. That is one way of doing. You can do PCR to find out. I will just use this space, or yes, this figure I will discuss in a while. Just follow the simplest ones, how to do that. We could have taken both of these plasmids. One is having a fragment, right? And one of it is not having a fragment. If the vector is about 5 kb and the fragment is about 2 kb, and this one is, the vector alone is having only 5 kb, we can run electrophoresis and we can figure out which one is, this will run slower compared to this one. We could have done restriction digestion, right? If we have done restriction digestion, then too we can figure out that this one would have formed one based on the, of course, based on the restriction endonuclease that we have used to clone in the first place. So it would have been unique site here. It would form one band, and the recombinant would have formed two bands, one of the vector and the other of the fragment, right? Then we know that is the recombinant one. We could have done PCR. How could we do PCR? We could have designed primers that are vector specific, okay? And there should be adjacent to on the either side of the cloning, multiple cloning site, which would also bind like this. If we performed PCR and we have run the products on electrophoresis, we would find the one without the fragment will be smaller, and the one with the template with the fragment will give a larger product. And that is another way of how we can find out which ones are recombinants and which ones are non-recombinants. Okay. The same way we can use many similar applications of what we have discussed. So we can take the recombinant, digest DNA from the recombinant clone. This will be the marker. This is vector, that is the ligation which we use to make the clone. And then we have taken the clone here and then digested it as well. This will form the vector this way, and it might give several inserts. And then we could have done further base if we already know the sequence of it. And here, assume if we have done a PCR, a cDNA hypothesis, cDNA library, for example. I think this is for general ways of how we do it normally. We can do clone 1, clone 2, clone 3, like that. These are controls. Marker is for trying to know the sizes. Vector is for control, so that we know which one is the vector fragment. And then we can use clone 1, clone 2, clone 3, clone 4, clone 5, like that. In the previous slide, previously when I was describing, I was only describing one example. Here you could have tested many colonies, not just one of it. And then you would try to find out which of these clones, okay, which of these clones is actually has the fragment of my interest. So the marker, as usual, will give numerous bands. The vector will give one band, and upon digestion, of course. And each of them will form a vector band like this. And they may have some bands like this additionally. And we can perform southern blotting using these, and using a probe that is of interest to our interest. And then we can figure out which of the clones has, say for example, this one gave the signal. Then we know that C4 is the clone that has the gene of interest in the cDNA library I made, or genomic library, based on your needs. Okay, that is how we can figure out the clone of interest among the cDNA library or genomic library. I thought I'll finish more. We have many more screening methods to discuss. This is important step in trying to solve many of the problems which we will discuss in the next class. Okay, I think I'll get to this in the next class. Do you have any questions? Any questions about the assignment? Or I hope I addressed the point of assignments in the last class. If you have any questions, please let me know. Any date fixed for CIA 2?
So we're discussing about it, sir.
Okay, when can you people let me know?
Within this night, possibly, sir.
Okay, by tomorrow you can tell me. Yeah, please discuss and let me know.
Yes, sir.
Sure, sir.
Okay, thank you then. If you have any questions, you can ask. Otherwise, you can move on to the next classes.